Imagine a computer that doesn't run on silicon chips and doesn't need traditional electricity. A system that actively repairs itself when damaged, operating entirely on biochemical energy. Imagine storing hundreds of petabytes of data inside a structure smaller than a grain of rice. This isn't distant science fiction. It is the emerging reality of biological computation. While artificial intelligence continues to push silicon hardware to its absolute physical limits, another quiet revolution is unfolding in molecular biology laboratories across the globe. Instead of transistors switching between zeros and ones, scientists are programming biological strands. They are engineering living bacteria and designing molecular circuits capable of processing complex data. The future of computing may not simply be inspired by living organisms. It may literally be alive. At its core, every living cell on Earth is already a sophisticated information processor. Inside a single human cell, millions of molecular interactions take place every single second. DNA stores instructions, RNA transports critical messages, and proteins execute complex operations. Countless biochemical pathways continually regulate decisions like growth, cellular repair, and movement. Abstract away the underlying chemistry, and the similarities to a digital computer are undeniable. DNA acts as hard drive storage, RNA behaves like temporary memory, and ribosomes function as processors. Cells receive inputs from their environment, process them through gene networks, and yield precise outputs. In many ways, evolution built programmable molecular computers billions of years before the transistor. Biological systems naturally operate in a massively parallel fashion without needing GPUs. Perhaps the most fascinating aspect of biological computation is the structure of DNA itself. The human genome stores 3 billion base pairs using 4 chemical symbols: A, T, C, and G. Researchers eventually realized something extraordinary. If DNA stores biological code, why not digital files? Today, scientists have successfully encoded books, images, and videos directly into synthetic DNA strands. They have even stored entire computer operating systems and scientific databases inside genetic material. The storage density of DNA is almost unimaginable compared to modern hard drives. A single gram of DNA can theoretically store over 200 petabytes of digital data. That means humanity's massive data centers could potentially fit inside a single coffee mug. Back in 1994, scientist Leonard Adleman used DNA molecules to solve complex mathematical problems. Instead of checking options sequentially like a CPU, millions of DNA strands calculated solutions simultaneously. Traditional software engineers write code in languages like C++, Java, or Python. Synthetic biologists write computational code using custom-engineered genetic sequences. Using synthetic biology, researchers create artificial biological circuits that mimic electronic logic. For example, a biological NAND gate produces a specific protein only when 2 target molecules are present. In a biological NOT gate, detecting a toxin causes the cell to shut down protein production. Scientists have even engineered bacteria capable of recording environmental events directly inside their genome. These living memories can record exposure to dangerous chemicals, radiation, or viral infections. Pushing boundaries further, researchers are constructing biological computers using real, living neurons. Lab-grown neural cultures have demonstrated remarkable abilities in adaptive learning and pattern recognition. These living neural networks suggest biological intelligence could complement silicon AI rather than compete with it. One of the most exciting frontiers for biological computation lies inside human medicine. Imagine microscopic biological computers traveling safely through your bloodstream every day. Instead of performing basic arithmetic, these microscopic devices monitor complex biological markers. If specific cancer markers and inflammation cross a set threshold, the cell triggers a targeted drug release. The medicine is delivered strictly to diseased cells, leaving healthy surrounding tissue completely untouched. This process requires no doctor intervention and no external electronic programming. Researchers are already engineering bacteria to detect early-stage tumors and sense internal inflammation. This autonomous biological decision-making promises to completely transform precision medicine. It moves healthcare away from generalized treatments toward real-time cellular computing. Despite immense promise, biological computation still faces critical engineering bottlenecks. Silicon processors operate at lightning speeds measured in gigahertz and nanoseconds. In contrast, biochemical processes unfold much slower, taking seconds or even minutes to complete. Living cells also exhibit natural biological variability and unpredictable mutation rates. Ensuring two genetically identical cells behave predictably under identical conditions remains difficult. Beyond technical limits, society will face profound ethical and biosecurity questions. Who owns genetically programmed organisms, and how do we prevent engineered systems from mutating? Safely integrating biological logic into ecosystems requires strict safeguards and multidisciplinary governance. The future of technology will likely not belong exclusively to silicon or biology alone. Instead, we are heading toward powerful hybrid architectures that merge the best of both worlds. High-speed silicon processors will handle rapid numerical calculations and AI model inference. Dense DNA strands will provide ultra-compact, long-term archive storage for human history. Meanwhile, living biological circuits inside our bodies will continuously monitor wellness and deliver therapies. Computation is not tied to silicon. It is a universal property of organized physical systems. The next great supercomputer won't hum in a server rack. It may grow in a lab and divide like a living cell.